Hello, this is Mitsuaki Sawano, Executive Associate Editor for JACC. I am very pleased to welcome today's guest and our very own JACC Associate Editor, Dr. Michael Honigberg, who is the corresponding author of two important papers being published as a simultaneous publication for the ESC meeting in Madrid. Dr. Honigberg is a cardiologist and director of research for the Women's Heart Health Program at Massachusetts General Hospital, assistant professor of medicine at Harvard Medical School, and an associate member of the Broad Institute of MIT and Harvard. His clinical focus is on heart disease prevention and women's cardiovascular health, and his research uses genetics, biomarkers, and large cohorts to better understand unique risk factors for cardiovascular disease. Welcome, Mike, and it's a pleasure to have you here with us today.
Thanks, Mitsu. Great to be with you today. Thanks so much for having me, for featuring these papers. We're really thrilled to be publishing these papers in JACC simultaneously with the ESC Congress.
Likewise. And we have two papers, both on clonal hematopoiesis of indeterminate potential, or better known as CHIP. Now, I have to confess, I have to admit that hematology has not always been my strongest area of expertise. So before we head into the two papers that we're going to discuss, could you tell our listeners what exactly CHIP is and why it's gaining attention in cardiovascular research?
Yeah, absolutely. It's a great question, and I suspect a lot of our readers and listeners probably aren't familiar with this form of CHIP, which is to say clonal hematopoiesis of indeterminate potential. So, a little bit of brief background on CHIP to set the stage for these papers. We know that our bodies acquire mutations across all of our tissues with aging, and most of them are of no clinical consequence. But a subset of mutations that our tissues acquire leads to what's called a proliferative advantage, and that they cause that cell to survive and expand. And that's the precursor state to cancer. We also know that aging is strongly associated with multiple chronic conditions, including cardiovascular disease, and that the actual mechanisms by which aging promotes disease risk beyond just accumulation of conventional risk factors are also not entirely understood. So about a decade ago, this prompted several different research groups to look across the population for the prevalence of these mutations that have been linked to the future development of blood cancer in circulating blood cells in apparently healthy people. And they were really surprised to find that the prevalence of these mutations, these clonal hematopoiesis of indeterminate potential mutations, was actually surprisingly high in apparently healthy people, with prevalence in excess of 10% in folks by the time they reach age 70. And as you would expect, because these are mutations in blood stem cells linked to the future development of blood cancer, the relative risk of blood cancer in CHIP carriers is pretty high, but fortunately blood cancers are rare, and most people who have CHIP mutations don't go on to get blood cancer. But a surprise finding of some of these early studies was that people who had CHIP mutations still had worse health outcomes and had worse mortality, and a lot of this excess mortality signal appeared to be driven by cardiovascular disease, which was a totally surprising and unexpected finding at the time. And since then, over the past decade, work by many groups, including our group, has sort of confirmed and helped to define what exactly those cardiovascular consequences are. And we are starting to understand the why. Why do these mutations in blood stem cells promote cardiovascular disease? And then what the heck do we do about it with respect to sort of prevention and treatment for our patients? I think one other point of background I'll just share is that this is, again, a pretty complicated area, really historically considered the realm of hematology and oncology. There are many, many different types there are many, many different genes, I should say, that can be mutated that lead to the condition of CHIP. And historically, just sort of due primarily to sample size and power, and also sort of evolving understanding of the field, historically, papers often lumped all of those types of chip together as one's composite entity, but we increasingly appreciate that chip is actually very heterogeneous, and we'll get into this, I think, with some of the results of these papers, but each mutation actually can behave quite different biologically and may have different implications for cardiovascular disease risk. And so I think that's sort of an emerging understanding in the field as well. The final thing I'll say that has sort of motivated some of this work is that even though now there's sort of a fairly robust body of literature demonstrating these associations, the magnitude of these associations over the previous literature has varied very widely. And there's still some, I think, some uncertainty or skepticism about whether these really represent a true causal link or whether this is sort of a marker of residual confounding or non-specific aging. And so that was some important motivation, I think, for some of the analyses that we're talking about today as well.
Thank you, Mike, for that great explanation on CHIP. So let's just dive into our first paper then. So the first paper, titled Clonal Hematopoiesis in Cardiovascular Outcomes in Older Women. Now, what was the question that you wanted to answer through this question for this paper?
Two key questions were top of mind we looked at a few, but I think the key thing we really wanted to understand in this analysis is CHIP still independently associated with adverse cardiovascular events in the elderly population? And if so, which types? And the background and motivation for that question is that some of the previous literature has actually cast doubt on whether CHIP remains an independent risk factor into later life. This is based on some prior work from our group as well as other groups, generally suggesting that the magnitude of associated risk seen with CHIP is largest in sort of midlife adults, midlife to late midlife adults, and then attenuates with age. And that's important because both CHIP and cardiovascular disease are highly aging related. And so understanding whether this aging related risk factor remains relevant into later life is really important for really understanding the cardiovascular disease relevance of CHIP overall. I think secondarily, a key opportunity in this analysis was to ask a sort of second related question, which is, does the type of sequencing that we use matter for understanding these relationships? And a little bit more brief background, historically, CHIP was detected in these sort of population-based cohort studies using technologies like whole genome and whole exome sequencing, which are great and help you answer a lot of questions, but their sensitivity for detecting CHIP, especially CHIP at sort of relatively smaller clone size, was modest because they're only sequencing so many copies of the genome in the peripheral blood. Increasingly, we have access to these very deep sequencing technologies that sequence the genome of blood cells, hundreds to thousands of times, which provides much more sensitivity for detecting CHIP and much more precision to quantify CHIP. The last thing I'll say before I turn it back over to you is that historically, CHIP was defined based on a fraction of mutated genes in the peripheral blood of greater than or equal to 2%. And that was really based on the sort of sensitivity of these historical sequencing approaches. But now with these deep, high coverage targeted sequencing platforms, we can detect CHIP down to smaller clone sizes, 1% of circulating blood cells or fewer. So we secondarily looked into that and whether associations varied across different thresholds for defining CHIP. But this was an opportunity to detect CHIP in this study cohort with much higher precision and fidelity than traditional approaches.
Great. Great to hear that there's advancement towards detecting these CHIPs. Mike, so what was the main finding from this paper?
I think to summarize our findings into three main findings. The first finding was that CHIP was indeed independently associated with cardiovascular events in this cohort, and I should give a little bit more context. This is a cohort of women who provided blood samples at a median age of 80 years. So this was really a sort of later life cohort of about 6,700 women who participated in the Women's Health Initiative Long Life Study. So a study of sort of longevity, healthy aging, and disease. So these are women who enrolled in the original Women's Health Initiative in the mid 1990s, and then participated in this follow-up study where they gave repeat blood samples, repeated study assessments, and then were followed for outcomes. So in this sort of much older group, again, median age 80 at baseline, CHIP did indeed associate independently with cardiovascular events. I think the second key finding, though, and sort of related to something I mentioned earlier, is that there appeared to be quite a bit of heterogeneity across these associations, both with respect to the key CHIP driver genes, so key subtypes of CHIP, and also with respect to the specific cardiovascular outcomes. So the three most common types of CHIP in the population, and this gets to be an acronym soup very quickly, but the three big ones are DNMT3A, TET2, and ASXL1. And those genes together represent about 80% of CHIP in the population. And of those three, prior literature suggests that TET2 is sort of the worst actor when it comes to adverse cardiovascular events. And so what we saw in our study here in the Women's Health Initiative Long Life Study was that when we looked at CHIP as a broad, when we lumped all the CHIP genes together, we actually didn't see any associations. So CHIP treated as a broad composite was negative. But it was when that we looked at each type individually is where we started to see the signals. And so TET2, again, the second most common type of CHIP in the population, TET2 CHIP, was associated, for example, with both incident coronary heart disease and with heart failure with preserved ejection fraction, or HFpEF. Both of these replicate previous signals, but now in this much later life cohort. ASXL1 had largely negative associations, with the exception of heart failure with reduced ejection fraction, or HFrEF, replicating a signal previously observed in the UK Biobank in their cardiac MRI cohort. Finally, the last gene that sort of emerged was JACC2. Clinicians have probably heard of JACC2 in the context of conditions like polycythemia vera and essential thrombocytosis. JACC2 was associated with several adverse outcomes, including venous thromboembolism, as you would expect, as well as cardiovascular death. So I think, the third related key finding is that CHIP is not only associated with coronary heart disease, which is sort of the best described association to date, but also with several other types of adverse cardiovascular outcomes beyond and independent of its link with atherosclerosis.
Thank you, Mike. And I just want to shift gears to our second paper. It's not usual to have two papers from the same corresponding author. I just want to make sure we have enough time. So the second paper is on LODOCO-2 study. So this is colchicine and longitudinal dynamics of clonal hematopoiesis, an exploratory substudy of the LODOCO-2 trial. So Mike, again, what was the question that you wanted to answer through this this paper? And maybe we could just briefly talk about the LODOCO-2 trial before we dive into the specifics.
Yeah, absolutely. So the first paper was the paper we just talked about was focused really more on understanding cardiovascular implications of CHIP. This paper is more now orienting toward potential therapeutic implications and what do we do about this condition. And before I describe the LODOCO-2 trial, I'll just give a little bit more background that motivated this paper, which is that several different lines of preclinical evidence have recently suggested that the growth of CHIP clones is partly driven by inflammation. That inflammation promotes the expansion, the selective expansion or the relative expansion of these mutant clones. And that's important from a disease standpoint because we know from the previous literature that larger clones are much more strongly linked to both adverse cardiovascular outcomes as well as to cancer risk. And so understanding drivers of clonal expansion could be important from a prevention standpoint, prevention of CHIP-associated chronic diseases, including both cardiovascular disease and cancer. Now, the LDOCO-2 trial provided a unique opportunity to test whether anti-inflammatory therapies might alter or modulate the growth of CHIP clones over time. So, by way of brief background on LDOCO-2, LDOCO-2 was a cardiovascular outcomes trial testing the effect of low-dose colchicine in a population with chronic coronary artery disease, enrolling approximately 5,500 individuals in the Netherlands and Australia. The primary trial findings were published in the New England Journal of Medicine in 2019 and indicated 31% reduction in a primary MACE endpoint in individuals randomized to colchicine compared with placebo. Now, LDOCO-2, we should sort of acknowledge at the outset, was a fairly pragmatic trial done on a relatively tight budget. And so there wasn't sort of trial-wide systematic biobanking of all participants at all time points. But a subset of sites in the Netherlands conducted a biobanking substudy where they collected blood samples at up to 4 time points, which included baseline prior to a 30-day colchicine run-in period, to after that 30-day colchicine run-in period, which was open label where everybody got colchicine. Then there was randomization following the 30-day open label run-in, and then blood samples were subsequently collected at 1 year after randomization, and then at the end of study, which was around a median follow-up of 25 months. And so, in this study, we tested the effect of randomization of colchicine on trajectories of CHIP clones in the LDOCO-2 trial population.
Great. And Mike, what was the main finding from this paper?
Our hypothesis was that randomization to colchicine, by modulating inflammatory signaling, we think primarily via the NLRP3 inflammasome pathway, although we know also by other pathways, we hypothesized that would blunt the growth or maybe even cause regression of CHIP clones in the group allocated to colchicine. And what we found was that there was, in the group randomized to placebo, we observed significant clear clonal growth across the randomized follow-up period. For the group randomized to colchicine, we saw substantially blunted growth, and many of the changes in clone size during follow-up were actually non-significant in the colchicine group, in contrast with placebo, where we saw a clear increase in clone size over time. When we looked at overall CHIP, and again, this gets to some of the heterogeneity we were talking about with the last paper, when we looked at overall CHIP, there was no significant interaction between time and randomization to colchicine, which is to say the differences in clonal growth trajectories did not differ statistically, sort of strictly speaking. When we dove deeper into key subtypes of CHIP, again, this TET2 CHIP sort of keeps emerging as one that's really important and might be particularly sort of pro-inflammatory. And for TET2 CHIP, we observed sort of statistically significant effect modification by randomization status, whereby the folks randomized to colchicine saw less growth of TET2 clones over time compared to folks randomized to placebo, which stated otherwise, colchicine seemed to significantly blunt the increase in TET2 clone size over time. So that was, I think, the main finding of this analysis. We also secondarily took advantage of inflammatory biomarkers that had been quantified in many of these same participants. And so we looked at the change in inflammatory biomarkers over time, stratified by CHIP status. The 2 markers that were available in the trial cohort were high sensitivity C-reactive protein, sort of a widely known blood biomarker of relative, non-specific inflammation or less specific inflammation, and then we also had the IL-6 level. So IL-6, a little bit more of a sort of a targeted inflammatory cytokine that's been more strongly linked to adverse cardiovascular events, with human genetics experiments sort of suggesting a causal role in accelerating cardiovascular disease. So we had both of those biomarkers over time, and the key finding was that for types of CHIP driven by mutations in genes other than DNMT3A, so sort of like all the other types other than that most common driver, colchicine very substantially reduced IL-6 levels over time compared to placebo. And that may be important, as we think about mechanisms of residual inflammatory risk in this population, and also some of the ongoing cardiovascular outcomes trials of therapies that are, selectively modulating IL-6 and IL-6 signaling. So, I think to summarize, colchicine appeared to reduce the increase in this particularly inflammatory type of chip called TET2, and colchicine seemed particularly, IL-6 levels seemed particularly modifiable by colchicine in a key subset of chip carriers.
Thank you, Mike. I mean, thank you for submitting these two exciting papers to JACC. I mean, this is kind of like a overarching question. So where do you see the field of like chip research heading in the next few years, and how could detection of chip eventually change clinical practice?
Yeah, I think those are really the key questions that emanate from these papers. And I think for now, chip is primarily in the research realm. And so I think with these findings in hand, where we need to be looking next is really to understand what the true therapeutic implications of harboring chip. And some things that we hypothesize based on these and other data might be that we might actually do things differently in chip carriers. This is a population that might be particularly responsive to anti-inflammatory therapies, either for primary or secondary prevention. We need to do a little bit more work to define exactly what the therapeutic implications of each individual chip subtype are. I think for TET2, there's a lot of interest in sort of modulation of inflammation for that one. For other subtypes, like for JACC2, preclinical data suggests that that's a group that might derive particular benefit from preventive aspirin use, even though as the field moved away from widespread aspirin use. So I think what's really needed next is prospective clinical trials that are both looking at effects of therapy stratified by chip status and or uniquely enrolling patients with chip and tested in targeted therapeutic strategies. I think we need, and I'm sort of saying all that from the cardiovascular standpoint. There's a whole parallel track of interest in this from the cancer standpoint, from prevention of progression to blood cancers and other forms of cancer, and so I think we need to demonstrate that there's actual clinical benefit and utility to understanding chip status before there's widespread adoption of screening or sequencing in apparently healthy individuals. But I think that's really the future direction for this line of research, is defining the translational impact and whether we can really improve outcomes by knowing somebody's chip status.
Mike, thank you again for sharing your expertise. And again, congratulations on these two important papers, simultaneous publications at JACC. And to our listeners, if you have the chance, please go say hi to Dr. Honigberg and his colleagues during the ESC. And thank you for joining us today.